Hi everyone, welcome to your lecture on DNA. DNA is deoxyribonucleic acid, and it is the common thread throughout life. Every living thing contains DNA. It's in the nucleus of every cell, and it is the instruction manual for how to make everything that is you. So the DNA contains the instructions for the cell to make the proteins, which help build your bones, your muscles, brand new cells, enzymes for doing chemical activities, and basically everything that your body needs. DNA was discovered recently, within about a little over 60 years ago now. And one of the people that was really important in the discovery of DNA was Rosalind Franklin. She was a scientist that was an X-ray crystallographer, which means she took pictures through the use of X-rays of the DNA molecule. And the photo that is pictured is photo 51, which is her most famous photo. And this is the photo that showed that DNA was in the shape of a helix, that it was helical and two-stranded. James Watson and Francis Crick were two other scientists working on discovering the shape and structure of DNA, and they were the first to build a model of DNA. And most of the time they were given credit for determining its shape. They did use help from Rosalind Franklin's research and her X-ray pictures in order to make their discovery. They named the shape the double helix. Double meaning there are two strands, and helix meaning that it is a spiral shape and it looks like a spiral staircase or a spiral ladder. So every single cell has DNA. In prokaryotic cells, which if you remember, prokaryotes are bacterial cells, the DNA is just located in the cytoplasm. It is not wrapped up in a nucleus because prokaryotic cells do not contain a nucleus. They have a single DNA molecule that is containing nearly all the cell's genetic information just right in the middle. And this next slide will show you a picture of it. So that is a bacterial cell, E. coli bacterium, and the blue string in the center of it is just the circular DNA. And again, it is not encased in a nucleus. It is just floating free in the cytoplasm. Many eukaryotes have 1,000 times the amount of DNA as prokaryotes. So eukaryotic cells are ones that do contain a nucleus, so they would apply to protists, fungus, plants, and animals. We are eukaryotic, and we have a lot more DNA than bacterial cells do. Eukaryotic DNA is located inside the nucleus. If you remember, the word eukaryote means true kernel or nugget or true nucleus. And the number of chromosomes varies widely from one species to the next. We have 23 pairs of chromosomes as humans, so 46 total. But many other species, plants, animals, fungus, they all have a very wide variety and number of chromosomes. So DNA is made up of many subunits or monomers known as nucleotides. Nucleotides are the monomer of DNA. They are considered the basic unit. And each nucleotide consists of three basic parts. You have this pentagonal sugar that is deoxyribose. DNA, the D stands for deoxyribose in the name. It's deoxyribonucleic acid, and the sugar is deoxyribose sugar. And it is the one that looks like a pentagon in the picture. It is labeled sugar, it's pink. The phosphate group is there in yellow, and the sugar and phosphates are all along the outside of the DNA molecule. And then the nitrogenous bases, there are 4 of them in DNA. They are adenine, thymine, guanine, and cytosine. And they are going to be on the inside of the ladder. And you see it pictured up in the top right corner. Nitrogenous just means it contains nitrogen. So you'll see throughout this molecule, the ends are nitrogens. So the 4 different bases are actually grouped into 2 separate groups. There are the purines, which are a double ring structure, and the pyrimidines, which are a single ring structure. So the purines are adenine and guanine, and pyrimidines are cytosine and thymine. So you always have a purine bonding to a pyrimidine, or a double ring structure bonding to a single ring structure. Okay, the way that I remember which ones are purines and which ones are pyrimidines are the following statements. Pure as gold. Pure as gold is for purines are adenine and guanine. And then pi cut. The pyrimidines are cytosine, uracil, and thymine. Uracil is a base that will show up in RNA only and replaces the base thymine. So uracil is for RNA, thymine is DNA specific, and we'll go over the differences between RNA in our next lecture. I also remember that when you get a piece of pie cut, it's a single slice. And pure as gold, I think of wedding rings. So when you have your engagement ring and the wedding band, there are 2 rings for females. And so I think of the pure as gold as the double ring and pie cut as a single slice of pie, single ring. Another interesting fact about purines is it was discovered when they were using urine, and so purines was actually pure urine. That's how they got the name. So the nitrogen bases always bond to the deoxyribose molecule, or the sugar of the nucleotide. You will always have the bases attached to the sugar. They will never be attached to the phosphate. The phosphate attaches to the sugar as well. So both the phosphate and the bases come out of the sugar. The sides or the rails of the ladder. If you describe DNA as a ladder type of structure, the rails are the sides that you would hold on to while you were climbing up of it. And it is deoxyribose and sugar and phosphate. So it'll be sugar, phosphate, sugar, phosphate, sugar, phosphate. And on the other side, it would be phosphate, sugar, phosphate, sugar, phosphate, sugar. They run in opposite directions. So the rails of the ladder are the sugar and phosphate, the ones that you would hold on to while you're walking up it. The rungs of the ladder, or the steps that you would put your feet on, are formed by the nitrogen bases. So they would be the adenine and thymine bonded together, or the guanine and cytosine bonded together. And they are bonded together with hydrogen bonds. This is a picture of that right now. You can see these dotted lines in between the adenine and thymine are the hydrogen bonds, and between the adenine and thymine, you see 2, which is there's a double hydrogen bond there, and between the guanine and cytosine, you see 3 lines, and those are the a triple hydrogen bond between guanine and cytosine. But A always bonds to T, and G always bonds to C. That was discovered by Erwin Chargaff, who was a scientist that came up with the pairing of the purines and the pyrimidines, that there was an equivalency between how much adenine there was and how much thymine there was, and between guanine and cytosine. That is because for every adenine, it bonds to a thymine, and guanine bonds to cytosine. So A to T and G to C. The way that I remember that is A and T together spell the word at. That's the only one that is a word. And G and C, the letters look very similar to each other. And if you think of the vitamin shops, also G and C, it reminds me of that. So I remember the word at and G and C. Just a couple tricks for remembering which ones go together. Again, this is just showing adenine always bonds to thymine, cytosine always bonds to guanine. This is Chargaff's rule. And adenine and guanine are purines, and thymine and cytosine are pyrimidines. So you always have a purine bonding to a pyrimidine or the double. The hydrogen bonds in the middle are very weak, and they're easy to separate. Hydrogen bonds are also what bond together water molecules, and if you remember from our unit of water, we talked about how they are a very weak bond, and they can be pulled apart very easy. So the 2 halves of the DNA molecule can easily unzip for replication purposes or getting transcribed into RNA. So the structure and the shape of the bases allow them to always bond to their complement, and the hydrogen bonds that hold them together are weak so that they can separate and come back together very, very frequently. The function of DNA is to carry the genetic information and to replicate it for future generations. All of our cells have to carry the genetic code, and we have to be able to pass it on to our offspring. And it carries out the orders to produce proteins necessary for biochemical reactions and overall life. It is also used to create enzymes. All of our cells contain the same DNA. They don't all do the same things with it. All of our cells are very specialized. So our skin cells will use different portions of the DNA code than, say, our blood cells will for creating the proteins and everything that they need to do their specialized job within our body. So this is 2 strands of DNA, and you should be able to tell me, just given one side of it, what the other side will look like. So if you push pause right now, you should be able to write down the 4 letters that would be the complementary to this first strand on the left side. Did you get them right? I hope you did. So remember that each nucleotide contains a sugar, a phosphate, and a base, and it is boxed here in red. That is a nucleotide. And you should take a moment to label the sugars with an S and the phosphates with a P and see if you notice a pattern. If you've done it right, you will have P, S, P, S, P, S, P, S on the left side, and then S, P, S, P, S, P, S, P on the right. And if you are looking for a pattern, you'll notice that the first one goes phosphate, sugar, phosphate, sugar. The next one is sugar, phosphate, sugar, phosphate. They are opposite of each other. They run in opposite directions. The order that the nucleotides are in, A, T, G, C, it determines which amino acids are used to make the proteins. And they make different words like the letters of the alphabet do when they're arranged in different structures or in different orders. I'm recording a video. I need you guys to be quiet. I'm going to have to edit you out. All of the DNA in your cell is 2 meters long, every single one of your cells. And the nucleus of your cell is only 2 micrometers in diameter. So that is a lot of DNA in a very, very, very small space. And so how does it all fit? Well, the answer to that is supercoiling. First, the DNA will wrap around histone proteins. So the DNA is a mixture of the deoxyribonucleic acids and proteins. It's not just DNA, it's also proteins. And it will wrap around the proteins, and this little green glob here is a histone protein. And then those proteins will wrap around each other. So the DNA and the histone together are called a nucleosome. And that is shown in this second picture. And then all the nucleosomes will coil up into what looks like a telephone cord, if you remember what corded telephones look like. But supercoiling is even farther than that. It's taking all of those coils and coiling them on top of each other. So if you remember corded telephones, they had the long coil of cord, and then sometimes that coil would then get wrapped on itself and get all tangled up. So the coil would coil again, and then it would coil again. So all of that coiling is going to super condense and make it really, really tight. And this is what DNA does before it becomes, when it is becoming a chromosome. So in the form of chromatin, it is very long and skinny and stringy. And when it supercoils up and gets really condensed, it is a chromosome. So that's just tons and tons of DNA packed very tightly together, really, really coiled up. And it does that before the cells divide during mitosis or meiosis. The cells will condense into chromosomes through the use of supercoiling. Here is a picture of a telephone cord, what I was showing you before. Just there's the one coil of all the nucleosomes coiled around each other, and then it can coil on itself again, and that would be a supercoil. And it does this multiple times. So what would a chromosome be? I hope you answered supercoiled DNA. And here you go, picture of it going from just the double helix, the ladder, all the way into a chromosome. Have a great day, and we'll talk again next lecture.